Bismillahirrahmanirrahim. Assalamu alaikum, dear students. I hope everyone will be fine and taking good care of herself. In our previous lecture, we had done this structure and function of cell wall, structure and function of plasma membrane. In today's lecture, we will do what are the cell surface markers, how the cell regulate its environment, what is cytoplasm and what are cell organelles. Then structure and function of endoplasmic reticulum, ribosomes, Golgi bodies and lysosomes. So the first topic is cell surface markers. Remember, in the plasma membrane on its outer surface, proteins and lipids, they are attached to the carbohydrates. If these carbohydrates are attached to the proteins, these are called glycoproteins, and if these are attached to the lipids, they are called glycolipids. These glycolipids and proteins, they act as cell surface markers or tags. These are specific in nature, means each and every cell have its own specific type of glycoproteins and glycolipids. So these are used for the recognition of the cell, and these are also used for the sticking of the correct cells when they are making the tissues and then organs and organ systems. So these glycoproteins and glycolipids, they act as cell surface markers, and each and every cell have its own specific markers or tags. These are different from one cell to the other cell. How cell regulate its internal environment? This is mainly through the transport, and transport occurs to obtain nutrients, to excrete waste. During the metabolic activity of the cell produce certain waste. This waste is required to be removed out of the cell, so that is also through transport from inside to outside. Similarly, during metabolic reaction, some products are produced. Some of these products are used inside the cell, but some are required to be used outside the cell, so they have to be secreted out. So these secretions, they are also released outside through the plasma membrane. Then for metabolic activities, certain ions are required, and these ions are required in certain concentration. Sometimes this concentration is higher as compared to the outer environment of the cell, and sometimes this concentration is low as compared to the outer environment. So it is a function of plasma membrane to maintain that concentration of the ions and the molecules inside the cell. Similarly, another function of the plasma membrane is to maintain pH. pH is important for the activity of the enzyme. You know that enzymes, they require specific pH for the function. So if this specific pH is there in the cell, then it would be active. That's why the cell, it maintains its pH through plasma membrane. Then how all these functions are maintained? This is through the semi-permeable nature of plasma membrane, where the molecules which are soluble in lipids, they can easily cross through it, but the molecules which are not soluble in lipid, they cannot cross through the plasma membrane. Remember, the main framework of the plasma membrane is made up of this lipid bilayer. So the molecules which are smaller in size and neutral, for example, oxygen and carbon dioxide, water and glucose molecules, they can easily cross through the plasma membrane. But the molecules which are not soluble in lipid, they cannot cross the plasma membrane. And similarly, the ions or larger molecules which are not soluble in lipids cannot cross the plasma membrane. So they have special proteins in the plasma membrane which have channels or gates through it. We had seen that in the previous lecture. So through these channels and the carriers, these ions and molecules, they are transported from outside to inside, or maybe from inside to outside. So this is to maintain the concentration gradient, and these proteins are present in the plasma membrane. And which type of movements are there for the movement of molecules through the plasma membrane? Basically, there are two types of movement: passive transport and active transport. Passive transport is that which do not require energy. It is of two types: diffusion and osmosis. Diffusion and osmosis is that where the molecules from the high concentration move towards the low concentration. That is according to the concentration gradient. That is from higher side towards the lower side. Difference between the osmosis and diffusion is that in diffusion solute molecules move from higher concentration to low concentration, but in case of osmosis, solvent molecules, they move from higher to low, and in most of the cases the solvent is water. And there is another type, this is facilitated diffusion. This is the movement of that ions we just have seen, which are not soluble in lipids, so they have to move through the channels. But they also move according to the concentration gradient from high towards the low. So this is facilitated by the proteins. That protein may be a channel protein or maybe a carrier protein, so this is called facilitated diffusion. All these are the types of passive transport. The second one is active transport. In the active transport, the movement is against the concentration gradient, that is from low concentration towards the high concentration. So to move the molecules or to push the molecules against their concentration, energy is required. This energy is usually provided by ATP. So this type of movement where molecules move from low towards the highest, this is called active transport. There is another type of transport that is called bulk transport. Bulk transport is that where the large particles which cannot cross the plasma membrane, they are obtained or they are taken in the cell in the form of vacuoles or vesicles. That's why it is called bulk transport, because large size molecules are taken inside the cell. It is again of two types, exocytosis and endocytosis. Exocytosis is that where something is released from cell to outside, and endocytosis where something from outside is taken inside the cell. Endocytosis is again of two types, that is phagocytosis and pinocytosis. Phagocytosis is that where the large solid particle, which is usually a food, is taken through the cell surface or through this plasma membrane. As you see, as soon as this food particle comes into contact with the plasma membrane invaginates and then surrounds that food and makes the food vacuole, and then it enters inside the cell. So here the large solid particle is taken inside, so it is called phagocytosis. In this case, liquids, these molecules, they are taken inside just like phagocytosis. That molecules, they are surrounded by plasma membrane and make a vacuole or a vesicle. This is called pinocytosis. So when the solid particle is taken through the plasma membrane, large solid particle, this is called phagocytosis. This is another figure that shows phagocytosis and pinocytosis. You can see that in phagocytosis, when large solid particle, that may be a food particle, comes into contact to the plasma membrane, it is engulfed by the plasma membrane and pinched off from rest of the membrane to make up a food vacuole, which is also called phagosome. And in case of pinocytosis, liquids or water molecules are taken inside the cell in the form of vesicle. This video shows the active and passive transport.
The smallest unit of life is the cell. In order for life to be sustained, nutrients and gases must be able to pass into a cell. Wastes and other products must be able to pass out of the cell. Many of the materials that pass in or out of the cell do so by passive transport. In this process, there is no energy, no ATP expenditure by the cell. Concentration gradients allow for the movement of materials. Osmosis and dialysis, both specific types of passive transport, use this concentration gradient of high to low. Another type of passive transport is facilitated diffusion. Since it is passive, the high to low concentration gradient is present. However, the process is facilitated by a carrier protein. The carrier protein is somewhat like a revolving door, allowing easy access across the membrane and then rotating back to the other side. All of these processes, osmosis, dialysis, and facilitated diffusion, are passive. The cell does not have to use any energy to move materials by these methods. The concentration gradient is used. However, there are materials that need to be moved against their concentration gradient. This process requires energy, ATP, and protein carriers that are found in the plasma membrane. Defined active transport is material moved from a low concentration to a high concentration through a membrane using a protein carrier that requires ATP. An example of active transport is the mechanism of uptake of iodine by the thyroid gland. Iodine is essential for the manufacture of hormones that influence body metabolism. When you eat a meal containing iodine, the iodine will pass from the small intestine into the circulatory system. As the capillaries pass through the thyroid gland, iodine diffuses out of the circulatory system. Iodine attaches to protein carriers in the thyroid cell's plasma membrane. Being proteins, they have a unique shape, so only iodine will fit. Glucose, which is also actively transported into the cell, relies on a different protein receptor. The energy required is somewhat analogous to a rock being pushed uphill. The processes of simple diffusion, osmosis and dialysis, facilitated diffusion, and active transport allow for most all of the small material, those dissolved in plasma and extracellular fluid, to pass in or out of the cell. Large particles, however, pass into the cell by endocytosis. This is an active process. It needs ATP. In this process, the plasma membrane surrounds the particle, such as bacteria or a food morsel, and forms a vesicle that pinches off from the membrane. The cell does this in two different ways. One way is called phagocytosis. In this method of cell eating, large particles are surrounded when the cell extends the plasma membrane and engulfs the particle. The result is a membrane-bound package of material. Fluids that the cell wants to take in enter by a process called pinocytosis. In this type of endocytosis, the cell membrane sinks in, the material sinks inward, and the membrane pinches off. The cell uses energy for this movement. In both types of endocytosis, the cell ends up with a membrane-bound vesicle. The material in the vesicle still has to pass through the membrane in order to be used by the cell. In this case, the lysosome joins with the vesicle and digestive enzymes break down the material to particles small enough to pass through the membrane and into the cytosol. A specialized endocytosis called receptor-mediated endocytosis also occurs in cells. In this process, molecules of a particular substance, such as cholesterol, bind to receptors in the plasma membrane. The vesicle is formed as the membrane sinks in. Then a lysosome joins the vesicle and the digestive enzymes break down the substance, releasing it from the receptors. The substance diffuses into the cytoplasm, the lysosome reforms, and the vesicle, with now empty receptors, becomes part of the plasma membrane again. This allows for the cell to reuse or recycle the receptors. A function of some cells is to manufacture or synthesize materials for export. In many cases, this material is made up of large molecules like proteins that cannot pass through a plasma membrane. The process of exocytosis enables the cell to pass this material out of the cell. The cell is a very dynamic living entity. It is essential that materials move in and out of the cell. This is done by passive transport using a concentration gradient and active transport, endocytosis and exocytosis, using ATP.
Now the cytoplasm means cell organelles. Cytoplasm is a part that is present between the plasma membrane and the nuclear membrane, as you can see in this figure. This is a plasma membrane. Inside the plasma membrane, this region is cytoplasm, and then this is nucleus. Remember protoplasm? which is a living matter of a cell. So cytoplasm is one of the part of that protoplasm. Cytoplasm is present in both prokaryotic and eukaryotic cells. Only difference is that in case of prokaryotic cells, membrane-bounded organelles are absent, and fibers or filaments that make up cytoskeleton, that is also absent in prokaryotic cell. Chemically, cytoplasm is made up of 90% water and remaining are the bioelements which are essential for normal life. These elements may be small molecules and ions like salts, which are inorganic, sugars, amino acids for proteins, fatty acids for fats, nucleotides for DNA and RNA, vitamins for many different metabolic reactions, and dissolved gases, just like oxygen, which is required for cellular respiration, and after this respiration carbon dioxide is produced as end product. So all these are soluble in water. They make up true solution. But the large molecules like proteins, fats, etc., they are not that soluble in water, so they make colloidal solution. As you can see in this figure, this is a true solution where the molecules are completely dissolved in the solvent, but in this case they are in the form of large particles or colloids. That's why this is a colloidal solution. Physically, cytoplasm is of two types. The part of the cytoplasm which is just around the nucleus, as you can see in this figure, this darker part around the nucleus is in liquid form. It is less viscous, so this is called cytosol. Sol is from solution. But the part of the cytoplasm which is towards the periphery, just inside the plasma membrane, in this figure it is shown in this light green color, it is gelatinous, jelly-like and viscous. So it is called cytogel. And you can see that most of the organelles, they are present in cytosol. Another feature of cytoplasm is that it can move. It moves in a circular manner. This is called the streaming movement of cytoplasm, and this movement is possible due to the filaments which make up cytoskeleton. When they contract, the cytoplasm moves. This movement is also called cyclosis. Due to this cyclosis, organelles can move from one place of the cell to the other place. Especially as you can see in this figure, the chloroplasts, they are also moving due to the cyclosis or streaming movement of cytoplasm. The purpose is that chloroplast moves to that site where they can capture maximum sunlight for their process of photosynthesis. Now, what is the function of cytoplasm? Cytoplasm is a site where most of the metabolic reactions are going on, because it is made up of many organic and inorganic molecules. So, and it is also the storehouse of the cell. The metabolic reactions which are going on in the cytoplasm, for example, protein synthesis, that is with the help of ribosomes. Glycolysis, it is the first step of cellular respiration that occurs in cytoplasm, as you can see in this figure. Glycolysis takes place in the cytosol, and then the end products of glycolysis are moved to mitochondria, where rest of the reaction takes place, and ATP is produced, which is used for many different functions. Glycogenolysis means glycogen breakdown. Glycogen is a complex molecule made up of many glucose molecules. It is an energy storage molecule. So whenever energy is required, glycogen is broken down to release energy. And gluconeogenesis means the formation of new glucose molecules from proteins, fatty acids, or from any other source. That occurs in cytosol. But the storage of useful and waste compounds, that take place in cytogel. The substances which are produced after metabolic reactions, they may be used by the cell, and some are required to be stored, so they are stored in the cytogel. And the waste which are produced in these metabolic reactions, they are required to be excreted out, but for the time being, before excretion, they are stored in cytogel. This is the streaming movement of cytoplasm in a plant cell. Now the cell organelles. In case of cytoplasm, there are two types of structures. First one is the non-living granules, which are actually the granules that have storage molecule or maybe the waste product which is required to be excreted out. They are not bounded by any membrane. They have no function to perform. They are called cytoplasmic intrusions. As you can see, this is a prokaryotic cell where these droplets of food are present in the cytoplasm. These are called cytoplasmic intrusion. Same is the case in eukaryotic cell, where these lipid droplets are the food granules or storage molecule. So these are cytoplasmic intrusions. The other main structure is cell organelle. Number of cell organelles are present in cytoplasm. These are bounded by cell membrane, and each and every organelle have a specific function to perform. For example, ribosomes, Golgi complex, mitochondria, chloroplasts, centrioles, endoplasmic reticulum, lysosomes, peroxisomes, glyoxysomes, and nucleus. Only ribosomes are the organelles which are without membrane. Otherwise, all these are surrounded by a membrane and have specific function. Ribosome also have a specific function. These are different organelles of cell. Now the first organelle is endoplasmic reticulum. Endoplasmic reticulum is a network of interconnecting tubules which are running throughout the cytoplasm from nucleus up to the plasma membrane. And these tubules, they are called cisternae. There are two types of endoplasmic reticulum: rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough endoplasmic reticulum is called rough because on its outer surface ribosomes are attached, so that appears rough in appearance. That's why it is called rough endoplasmic reticulum. But smooth is without any ribosomes, so the appearance is smooth. That's why they are called smooth endoplasmic reticulum. And you can see that both are present side by side. A specific type of smooth endoplasmic reticulum is present in the skeletal muscle, where it helps in the muscle in its function. It is called sarcoplasmic reticulum. This is another figure of endoplasmic reticulum. This is a nuclear membrane. To this nuclear membrane is attached rough endoplasmic reticulum. To its outer surface, ribosomes are attached. And then this one is smooth endoplasmic reticulum without any ribosomes. Now the rough endoplasmic reticulum, it is called rough because of the ribosomes attached to its outer surface. Its major function due to the attachment of ribosome is protein synthesis, because ribosomes are actually responsible for protein synthesis. So rough endoplasmic reticulum along with these ribosomes, they synthesize proteins. And as the endoplasmic reticulum extends from nucleus up to the plasma membrane and throughout the cytoplasm, so they also provide mechanical support. These proteins which are synthesized in endoplasmic reticulum, they are transported to the Golgi apparatus, where they are modified for many different functions. Smooth endoplasmic reticulum is smooth because it is without ribosomes, so its appearance is smooth. It has many different functions to perform. First of all, it is responsible for the metabolism of carbohydrates, for the synthesis of lipids, especially phospholipids and steroids. Remember, these are the major parts of plasma membrane. Another important function of smooth endoplasmic reticulum is detoxification of drugs and poisons. If any poison substance enters inside the body, it is detoxified or deneutralized by the smooth endoplasmic reticulum presence, especially in the liver cells. Another function of smooth endoplasmic reticulum, especially which is present in the skeletal muscle, it stores calcium. And calcium is very important for the contraction of skeletal muscle. And just like rough endoplasmic reticulum, smooth endoplasmic reticulum, it also provides mechanical support. And smooth endoplasmic reticulum helps this rough endoplasmic reticulum to transport proteins which are synthesized in rough endoplasmic reticulum to the Golgi apparatus. Now the next is ribosomes. Ribosomes are the only organelles which are not bounded by membrane, and they are present in both eukaryotic and prokaryotic cell. They were discovered in the electron microscope for the first time as granules, and they are spherical granular structures of about 20 to 24 nanometer in diameter in case of eukaryotes, and chemically they are made up of ribonucleoproteins, that is RNA plus proteins. Equal amount of RNA proteins make up ribonucleoproteins or ribosomes. And ribosomes, they are responsible for protein synthesis. These ribosomes, they attach with the messenger RNA. You can see in this figure, this blue strand, this is a messenger RNA which brings message from DNA for the synthesis of protein. This message is translated by the ribosome. So this then synthesizes protein molecule. Ribosomes are synthesized inside the nucleolus, which is present in the nucleus, and after its formation, these ribosomes are transported to the cytoplasm where they perform their function of protein synthesis, and they are transported from nucleus to the cytoplasm through nuclear pore. These are the pores which are present in the nucleus. As we just have seen that ribosomes are present in both prokaryotic and eukaryotic cell. But the only difference is that in case of eukaryotic cell, the ribosomes are larger in size and they are characterized as ATS. S is Swedberg unit. It is the unit for sedimentation rate, the rate at which these units or subunits, they settle down during centrifugation. Inshallah, we will do that in the lab whenever it will be possible. But in case of prokaryotic cells, ribosomes are smaller as compared to eukaryotic ribosomes, and they are characterized as 70S. Ribosomes are also present in both mitochondria and in the chloroplast. They are present in the matrix of mitochondria. You see in this figure, these dots, it is representing the ribosomes of mitochondria, and same is the case in the stroma of chloroplast. These ribosomes are present. It means that these organelles are also able to synthesize its own protein, which are required by these organelles for their function. But it is believed that the ribosomes present in these organelles, they are smaller as compared to the ribosomes present in the cytoplasm, and these ribosomes are of the size of prokaryotic ribosomes, that is 70S ribosomes. And ribosomes which are present in eukaryotic cells, they may be present either freely dispersed form in the cytoplasm, or they may be attached to the rough endoplasmic reticulum. Ribosomes are made up of two subunits, and both subunits are of different sizes. One is larger and the other one is smaller. In case of eukaryotic ribosome, the larger subunit is 80S and the smaller one is 40S. And when they combine together, that makes 80S. As you can see in this figure, that is the eukaryotic ribosome, where the larger is 60S, smaller is 40S. When they combine, they make 80S. When they are not synthesizing proteins, these two subunits remain separate from each other in the cytoplasm, but in the presence of messenger RNA, they combine together to start protein synthesis. Same is the case in prokaryotic ribosome. It is also made up of two subunits. Larger subunit is of 50S, smaller one is of 30S, and when these two subunits join together, it makes 70S ribosome. The union of two subunits is controlled by the presence of magnesium ions, or these two subunits may form a salt bond between the phosphate of RNA of one subunit and the amino group of amino acid of another subunit. So they join to make up a complete ribosome, which then helps in protein synthesis. And they attach together only when they are doing protein synthesis. And in many cases, one single strand of messenger RNA is attached by a number of ribosomes. so that each and every ribosome synthesizes the same protein. So at the same time, number of molecules of the same protein are synthesized. Such structure where one single strand of messenger RNA is attached by many ribosomes, this is called polysome. This is another figure that shows the two subunits of ribosome, larger one and smaller one. Here they are fused together during the protein synthesis.
Welcome to Moo Moo Math and Science. In this video, I'd like to talk about the ribosomes. When you look at pictures of cells, you can see tiny dots on the ER and in the cytoplasm. These dots are ribosomes. But why are they important to the cell? Ribosomes are small protein factories found in cells. They are located in the cytoplasm and on the rough ER. Ribosomes look like small dots on the ER and in the cytoplasm. They are found in plant, animal, and bacterial cells. How does a ribosome make this protein? First, messenger RNA that has been created at the nucleus travels to the ribosome. Transfer RNA travels to the messenger RNA and reads it in triplets called codons. The transfer RNA has amino acid on one end. The transfer RNA reads the messenger RNA, and a string of amino acids are put together in the correct order. Now, why is protein important? Protein is used for many functions in the cell. They are used as building blocks for structure, support. Your skin and tendons are made of proteins. Some proteins help with digestion. Proteins help with muscle contraction and also help carry oxygen around your body. So there we go. These tiny organelles called ribosomes are very important in helping the cell stay alive. Thanks for watching.
Now the next organelle is Golgi complex. Golgi complex is named after the scientist who discovered it, Camillo Golgi, in 1898. Golgi complex is made up of flattened tubules which are stacked one above the other to make up this pile of tubules which are called cisternae. These cisternae have two sides or two faces. One is convex, this is towards the endoplasmic reticulum. This is called cis phase or forming phase, and the other one is concave side, which is also called transphase or maturing phase. The vesicles which are coming from the endoplasmic reticulum carrying the protein synthesized by endoplasmic reticulum, they then fuse to the Golgi apparatus at the cis phase or the forming phase, and these proteins are modified inside the Golgi apparatus. After modification, they are released from the transphase or maturing phase in the form of vesicles. These vesicles then have secretions which are then released out. This is another figure that shows Golgi apparatus. You can see in this figure, it is very much clear that the cis phase is that where the transport vesicles, they come and fuse with the forming phase, release the contents in these cisternae of the Golgi where these secretions are modified, and then they are released from the transphase in the form of vesicles. These are also the secretory vesicles, may have secretions in it. Functions of Golgi apparatus, again same, you see in this figure. This is rough endoplasmic reticulum. This green is a protein or polypeptide synthesized in the rough endoplasmic reticulum. Then through these transport protein vesicles, it is transported to Golgi, which is also known as Golgi complex, Golgi body, Golgi apparatus, where they are modified. After modification, these molecules may be the secretions, and these secretions are inside the secretory vesicles. They are to be released out, and this is done by exocytosis. Golgi apparatus also gave rise to lysosomes. As you can see in this figure, The proteins which are synthesized in the rough endoplasmic reticulum are transported to the Golgi through this transport vesicle. After modification in this Golgi apparatus, these proteins may be the secretions which are then to be released out, as we had seen in the previous figure. But in this case, some of the proteins which are synthesized by the endoplasmic reticulum, they make up enzymes, digesting enzymes, which when released from the Golgi apparatus in the form of vesicles, they are called lysosomes. They help in the digestion of food which is taken in by the cell as food vacuole, and they are also used for the destruction of old and damaged part of the cell. So, and Golgi apparatus give rise to lysosomes, peroxisomes and glyoxysomes. Peroxisomes and glyoxysomes are also the structures similar to lysosomes. And the modification which takes place in the Golgi apparatus, it is that protein molecules may combine with another molecule, that may be a lipid or a carbohydrate, to make up a complex molecule that is called conjugated molecule. If protein combines with lipid, it is called glycoprotein. If protein combines with lipid, it is called lipoprotein. Sometimes the protein which is synthesized in rough endoplasmic reticulum, it is larger than the required size, so its cleavage also takes place in the Golgi apparatus, and then as an end product, it is released through the vesicles from the transphase. This is another figure. I think this is very much clear, which shows these transport vesicles that take the protein synthesized in endoplasmic reticulum to the Golgi apparatus, where these proteins may make up secretion. So they are bounded by a membrane to make up secretory vesicle, released from the transphase of endoplasmic Golgi apparatus, and then released out by exocytosis, or they make up proteins after modification. They make up lysosomes. Lysosomes help in the digestion of food vacuole which is taken in through phagocytosis. Another very important function of Golgi apparatus is the division of cytoplasm during cytokinesis, which is the division of cytoplasm during cell division. When cell is dividing, especially in the plant cell, where cell wall is very rigid, it cannot invaginate to divide the cell into two halves. So the vesicles from the Golgi, they line up at the center or equator of the cell. This is called phragmoplast. These vesicles contain the material for the synthesis of cell wall. Then these vesicles fuse together to make up cell plate, which gradually elongates, and then it make up a new cell wall for both new daughter cells. Now the next is lysosomes. Lyso means splitting and soma means body. It is discovered by D. Dube in 1949. They are bounded by a single membrane, spherical in structure, about 0.2 to 0.5 micrometer diameter. And lysosome is an organelle which is present only in animal cell. Their major function, as we just have seen, is that they have hydrolytic enzyme, means enzymes which can digest different substances or different molecules. And these enzymes inside the lysosome, they are present in acidic medium. So the pH of lysosome is usually acidic, from 4 to 4.5. And the enzymes of the lysosomes, they are synthesized in rough endoplasmic reticulum, and then they are transported to Golgi, where they are modified, and then they pinch off from the transphase of Golgi in the form of lysosomes. And these lysosomes, which are pinched off from Golgi apparatus, they are called primary lysosomes. Again, the same figure, where lysosomes are being formed from Golgi apparatus. They help in the digestion of food which is taken in by phagocytosis, and also damage the old and the damaged parts of the cell. The function of lysosome, as I just told you, as they have hydrolytic enzymes, so their function is to digest food, to destroy the old and damaged part of the cell. And how is this done? The primary lysosomes, they combine with different types of vesicles. These vesicles may be formed by endocytosis. Remember endocytosis? So the vesicles which are formed by endocytosis are called endosomes. If these vesicles are formed by phagocytosis, they are called phagosomes. And another type of vesicles are called autophagocytosis. Autophagocytosis is that in which the parts of the cell, which are present inside the cell, they are surrounded by a membrane. So that is called autophagosome, and the process is called autophagocytosis because this parts will be then destroyed. So these vesicles, which may be formed by endocytosis, by phagocytosis, or maybe by autophagocytosis, when these vesicles combine with the primary lysosomes, they make up secondary lysosomes. These secondary lysosomes are also called endolysosomes. So secondary lysosomes release their enzymes in the endosomes, phagosomes or autophagosomes and destroy and damage whatever particle or molecules are present in them. And these molecules which are digested in the secondary lysosome, they are converted into simpler form. If it is a food, of course, it would be converted into amino acids, monosaccharides, fatty acids. Even the old and damaged parts, their useful compounds would also be converted into simpler one, which can be reused again by the cell. This is a figure that shows three different types of vesicles which are formed by three different types of processes. As you can see, phagocytosis, taking in of solid particle. Here is a bacterium. When it is taken inside the cell in the form of a vesicle, this is called phagosome. It combines with primary lysosome to make up secondary lysosome, and the solid particles taken up by endocytosis. This vesicle which is formed by endocytosis is called endosome. It also combines with primary lysosome to make up secondary lysosome. Now autophagocytosis is that where the old and the damaged part, or maybe the organelle, as in this case it is a mitochondria, it is required to be destroyed. It is surrounded by the membrane which is synthesized by the endoplasmic reticulum, so it makes up a vesicle. This is called autophagosome, and this process of formation of this vesicle, it is called autophagocytosis. This autophagosome then combines with primary lysosome and makes up secondary lysosome. So secondary lysosome is also known as endolysosomes. Primary lysosome release its enzyme inside these phagosome, endosome, or it may be autophagosome, and digest the components or molecules present inside these vesicles.
Lysosomes are membrane-bound vesicles that contain hydrolytic enzymes. The hydrolytic enzymes degrade proteins, nucleic acids, lipids, and carbohydrates, and are formed in the endoplasmic reticulum. These enzymes are then transported to the Golgi apparatus by transport vesicles. The lysosomes arise from the Golgi apparatus. When particles such as viruses or bacteria are ingested by phagocytosis, the lysosome fuses with the particle-containing vesicle, called a phagosome, and delivers the hydrolytic enzymes. Lysosomes also fuse with organelles such as old mitochondria. This results in the destruction and recycling of these structures.
So the lysosomes are responsible for three major functions, that is intracellular digestion, autophagy and autolysis. First one is intracellular digestion. In this, as we just have seen, that food vacuole which is formed by phagocytosis, and it is called phagosome. When it is combined with primary lysosome, it makes up secondary lysosome. Enzymes of the lysosomes, they are released in this phagosome, and then these enzymes digest whatever material is present inside the phagosome, and these digested material, if it is useful, it is absorbed in the cytoplasm, and what is waste, it is then released out by exocytosis. And before it's released, the waste products inside these vesicles are there or stored for a time being. So this vesicle is now called contractile vacuole. This is another figure of intracellular digestion. As you can see, that phagosome formed by phagocytosis combines with the lysosomes which are produced from Golgi apparatus. Enzymes of the lysosomes are released in the phagosome. So these enzymes digest up the food particles. Digested food is absorbed in the cytoplasm, and the waste material is then released out by exocytosis. Next function of lysosome is autophagy. Auto means self, phagy means eating. Self-eating means destruction of the old and the worn out particles or cell organelles of the cell. And it is that when some parts of the cells, they are not required, they are old, and they are no more able to perform any activity in the cell, they then, as we already had seen, that they are surrounded by a membrane which is produced by endoplasmic reticulum to make up a vesicle that is called autophagosome. This autophagosome then combines with lysosome. Lysosome releases its enzymes inside it, and then it destroys these organelles. So these organelles, then the components or simpler molecules of these organelles are reused again, and the waste is excreted out by exocytosis. And usually this occurs when energy is required during starvation, when we are fasting. So this usually occurs at that time. And it also controls the number of organelles. For example, when we start heavy exercise, the number of mitochondria, it increases in the muscle cells. But when we stop exercising, these mitochondria, as they are extra no more required by the cells, so they are destroyed by this autophagy. Why the number of mitochondria increases? Because during heavy exercise, more energy is required. This more energy is required, or it is fulfilled by the oxygen which is taken in during respiration. So that's why the number of mitochondria increases inside the cell. Third function of the lysosome is autolysis, means breakdown of the cell. That's why these lysosomes are also called suicidal bags. Whenever the cell completes its lifetime and is then required to be destroyed or disintegrated, then lysosomes which have hydrolytic enzymes, these enzymes are released due to the bursting of the membrane of the lysosome in the cell, and these enzymes then start eating up the cell contents, so the whole of the cell is destroyed. This is also called programmed death, because it is the important feature during developmental process. During development, many different structures are formed which are later on in the adult stage are no more required. So these structures are destroyed due to this autolysis, or it is also called apoptosis. Now, the lysosomes which have hydrolytic enzymes, they then digest many different molecules inside the cell. If any of the enzyme present in the lysosome is defective, may not be able to perform its function, may be absent, or may not be able to perform a function, in that case, that particular molecule which is digested by that particular enzyme, it will start piling up inside the cell. So it results in diseases that are called lysosomal storage diseases. And these diseases are hereditary due to mutation in the genes and are congenital, means present at the time of birth. There are many different types of lysosomal storage diseases. There are about 20 different types, and one of the most important is that Tay-sachs disease. In Tay-sachs disease, actually, the enzyme which is responsible for the digesting of lipids in the nervous system is missing. So the lipids, they start accumulating in the nerve cells, so result in the damage of the nerve cells. And ultimately, the nervous system is also damaged, and usually this is present at the time of birth, and these individuals, they do not survive. So that is all about for today's lecture. This is another picture that shows a Tay-sachs disease, in which you see this is the normal lysosome that is digesting this lipid molecule with its enzymes. But in Tay-sachs disease, the lipids digesting enzyme is absent in the lysosome, so lipids start piling up in the nervous cell, resulting in the destruction of a nerve cell. Your assignment is that to draw a neat and labeled diagram of fluid mosaic model of the plasma membrane and plant cell wall. And number 2, write down structure and functions of endoplasmic reticulum, ribosomes, Golgi complex, and lysosomes.